Welcome to the Deep Dive. Today we're diving into some really fascinating science, looking at a new way, potentially, to treat liver disease caused by Alpha-1 Antitrypsin Deficiency, or ATD.
Yeah, it's definitely a complex area, but there's some exciting stuff happening.
Absolutely. We're going to break down what's important and, honestly, pretty cutting edge here.
Right. We're seeing this wave of therapies using RNA interference, RNAi, and it's looking particularly hopeful for, well, a big unmet need in AATD.
So maybe just a quick refresher on AATD for everyone.
Sure. So it's a genetic disorder basically stemming from mutations in the SERPINA1 gene, and often the Z-AAT variant is the one linked to the more severe cases.
Okay, SERPINA1, what's its day job in the body, normally?
Normally, yeah, it provides the instructions for making Alpha-1 antitrypsin. This is a crucial protein. Its main job is protecting tissues, particularly the lungs, from enzymes like neutrophil elastase.
Kind of like a bodyguard for the lungs.
Exactly. That's a great analogy.
Got it. And this EAT mutation, it messes that up somehow.
Precisely. The Z allele, specifically a mutation called GLU342 lyase, it causes the AAT protein to misfold as it's being made. And instead of being secreted into the blood to do its job, this faulty Z-AAT protein gets trapped. It accumulates inside the liver cells, the hepatocytes.
Oh, so it's like a double whammy then.
Yeah.
You don't get enough of the good AAT out protecting the lungs, which can lead to things like emphysema.
Right, the loss of function problem.
But today, we're really honing in on the other side of it, the liver. This buildup of the bad protein causing actual damage in the liver.
That's the gain-of-function toxicity, yeah. And current treatments, like augmentation therapy, they boost the good AAT for the lungs.
But they don't really do anything about the liver issue.
Exactly. That's where these new RNAi therapies are aiming, directly targeting the liver disease aspect AATD-LD. And that comparative analysis you shared really lays out this emerging field well.
So our mission for this deep dive,
Yeah.
really get into how RNAi works for AATD-LD, what makes these therapies different, why it's such a big deal, and where things stand right now.
Sounds good.
Okay, let's start with the science of the problem, maybe a bit deeper. The gene, the mutation, what's actually happening inside those liver cells when the misfolded protein piles up?
Right, so it's not just sitting there. These misfolded proteins, they tend to stick together, they form polymers. And these polymers clump up into these characteristic structures you can actually see under a microscope. They're called KASD globules.
Like KASD globules.
And this accumulation isn't passive. It puts immense stress on the cell's machinery, especially the endoplasmic reticulum, the ER, which is involved in protein folding and processing.
So the cell's quality control system gets overwhelmed.
Pretty much. Think of it like a factory assembly line getting jammed up. It disrupts normal function. It also seems to affect the mitochondria, the cell's power plants, leading to oxidative stress, and it triggers inflammatory signals.
ER stress, inflammation, mitochondrial problems.
Yeah, that sounds like a recipe for disaster at the cellular level.
It really is. And over time, this chronic injury and inflammation can lead to liver fibrosis that's scarring.
Scar tissue building up. Yeah, replacing healthy tissue. And if that progresses, it can lead to cirrhosis, which is severe, often irreversible scarring. And it also significantly increases the risk of liver cancer, hepatocellular carcinoma.
And this isn't just an older adult problem, is it? I remember the sources mentioning liver issues can show up in kids too.
That's a really important point. Some estimates suggest, I think, maybe 50 to 80% of children with the PZ genotype can show signs of liver disease, which just underscores the urgent need for therapies that target the liver directly. Because right now, for severe AATD liver disease, the main option is often a liver transplant.
Which is a massive intervention.
Huge, with all its own risks and challenges. So that brings us to RNAi. This is where the new hope lies for the liver side. How does this RNA interference actually work here?
So RNA interference, it's actually a natural cellular process. Cells use it to regulate gene expression, kind of like a dimmer switch.
Okay.
Therapeutic RNAi takes advantage of this. It uses specially designed molecules called small interfering RNAs, or siRNAs.
And these siRNAs are engineered to be highly specific. Think of them like guided missiles for messenger RNA, the mRNA.
The instructions copied from the DNA blueprint.
Exactly. In AATD-LD, the siRNA is designed to find and bind to the mRNA that carries the instructions for making that faulty Z-AAT protein, specifically within the liver cell.
And what happens when it binds?
It recruits the cell's own machinery, something called the RISC complex, which then essentially chops up or degrades that specific Z-AAT mRNA.
So you're stopping the production line before the faulty protein even gets made.
Precisely. Less mRNA means less production of the toxic Z-AAT protein. The goal is to reduce that buildup, ease the stress on the liver cells, and hopefully prevent or slow down the liver damage.
Makes sense. But how do you get these RNAi molecules into the liver cells effectively? Delivery seems like it would be tricky.
Yes, delivery is absolutely key. And this is where a really clever bit of technology comes in GalNAc conjugation.
Cool.
N-acetylgalactosamine. It's a type of sugar molecule. The RNAi molecules are chemically linked to these GalNAc sugars.
Okay. Why that specific sugar?
Because liver cells have a receptor on their surface, the asialoglycoprotein receptor, or ASGPR, that just loves to bind to GalNAc. There are tons of these receptors on liver cells.
Like a specific docking station just for liver cells.
Exactly. So when you inject the GalNAc-Serrana conjugate, usually subcutaneously just under the skin, it travels through the bloodstream, finds those liver cells, binds to the ASGPR receptors, and gets pulled inside. It's a highly targeted delivery system.
Which means more of the drug gets where it needs to go and less goes elsewhere.
Right. Much more efficient delivery to the liver. And this allows for potentially lower doses and, importantly, less frequent injections, maybe just a few times a year, which is a huge improvement for patients compared to, say, weekly or daily therapies. This GalNAc tech is really a game changer for RNAi liver targeting.
That targeted delivery sounds like a massive improvement. The sources mentioned some promising early results in mice, the PiZ mice.
Yes, the preclinical work was very encouraging. In those mice, which mimic the human disease quite well, the RNAi treatments significantly reduced the amount of those Z-AAT protein polymers in the liver.
Okay.
They also saw improvements in the liver cells themselves. They looked healthier. And some downstream effects, like reduced inflammation and even preventing liver tumors in some models, started to normalize.
That's pretty compelling proof of concept data. So let's move to the clinical side. The therapy that seems to be furthest along is fazirsiran.
Correct. Fazirsiran, it used to be called AROAAT, and Takeda refers to it as TAKE-999. It's being co-developed by Arrowhead Pharmaceuticals and Takeda.
And it uses this RNAi approach with the GalNAc delivery.
Yes. It uses Arrowhead's TriRME platform, which incorporates that GalNAc targeting tech we just talked about, delivered by subcutaneous injection, aiming to silence the SERPINA1 gene expression in the liver.
Okay. And what have the clinical trials shown so far? It's gone through phase 1 and phase 2.
It has. Phase 1 studies basically confirmed it could significantly lower the levels of AAT protein circulating in the blood in humans, and the safety looked good, supporting less frequent dosing.
Right. Then phase 2.
There were a couple of key phase 2 studies. One was an open-label study, ARA 2002. This showed really substantial reductions in Z-AAT, not just in the blood, but also measured directly in liver biopsy samples.
Actually measuring it in the liver tissue itself.
Right. And very importantly, they saw some encouraging signs in the liver histology, fewer of those PSE globules, and maybe even some reduction in fibrosis in some patients. That was an early, exciting hint.
A hint of fibrosis regression. Okay. But then there was a larger placebo-controlled phase 2 trial, Sequoia.
Yes, Sequoia was designed to confirm these findings more rigorously. It definitely confirmed the powerful effect on reducing Z-AAT levels, both serum and liver, consistently. It also showed improvements in those globules and signs of reduced liver inflammation.
Sounds good so far.
But the tricky part came with the fibrosis endpoint. While some patients on fazirsiran did show improvement in fibrosis over the year, so did a noticeable number of patients in the placebo group.
A significant placebo response on the fibrosis measure.
Exactly, which made it difficult to say definitively within that 1-year time frame how much of the improvement in the treated group was specifically due to the drug versus just, well, natural variation or other factors.
That is interesting. Does that imply some level of maybe natural fluctuation or potential for some reversal, even without specific treatment over that period?
It could suggest that, or perhaps limitations in how fibrosis is measured, or just the inherent variability of the disease progression. It really highlighted the need for a longer, larger study.
Which leads us to phase 3.
Right. The ongoing phase 3 trial is called Redwood. It's a large study following patients for a longer duration, I believe around 2 years, specifically looking at histological improvement, that is, changes in liver tissue, with a primary focus on fibrosis regression.
So, summing up fazirsiran's efficacy, strong, consistent reduction of the target Z-AAT protein in blood and liver, reduces the protein globules, seems to reduce inflammation. But the big question mark is definitively proving fibrosis reversal. pending those phase 3 Redwood results.
That's a fair summary, yes.
And what about safety and tolerability through phase 2?
Generally, it's been reported as well tolerated, no major safety signals popped up, and crucially, no negative effects on lung function were observed, which is always something to monitor when you're lowering total AAT levels.
That's got some regulatory recognition.
Yes, the FDA granted it breakthrough therapy designation, which could potentially speed things up if the phase 3 data are positive.
And the timeline for those results?
Yeah.
Still a few years out.
The estimates are pointing towards late 2026, maybe 2027, for the primary phase 3 results. If all goes well, a potential market launch could follow, perhaps in the 2028-2029 timeframe.
Okay, so fazirsiran is clearly the one to watch in the RNAi space. But it wasn't the only player, right? The sources mentioned another one, belcesiran.
That's right, belcesiran. It was developed initially by Dicerna Pharmaceuticals, ticker DCRA1AT, and then became NN6021 after Novo Nordisk acquired Dicerna.
And it uses similar approach, RNAi, GalNAc delivery.
Yes, very similar mechanism. Targeting the serpin-1 mRNA using RNAi delivered subcutaneously, leveraging Dicerna's GalXC platform, their version of GalNAc targeting.
Did it show promise in early studies too?
The phase 1 results looked quite good. They reported robust, dose-dependent reductions in serum AT, with some individuals reaching levels similar to what fazirsiran showed. It was also generally well-tolerated, no major safety issues flagged, no lung function concerns noted in that early data.
So sounded like another viable candidate. They started a phase 2.
They did. Right. We're seeing this wave of therapies using RNA interference, RNAi, and it's looking particularly hopeful for, well, a big unmet need in AATD.
But that trial didn't complete.
No, it was terminated, and unfortunately no data from the Estrella phase 2 study was ever publicly released.
Any idea why it was stopped?
The most likely reason points towards a strategic portfolio decision by Novo Nordisk after they acquired Dicerna. It's pretty common post-acquisition for companies to reassess pipelines.
Right.
Given that fazirsiran already had a significant lead in development, Novo might have decided to focus their resources elsewhere rather than compete directly in the same space with the later stage asset.
So a business decision rather than necessarily a scientific or safety issue with belcesiran itself, as far as we know.
That seems to be the most probable explanation based on the available information. So belcesiran, while initially promising, is off the table now.
Okay. That certainly highlights the competitive landscape and, I guess, the high stakes involved. Let's zoom out then. Beyond these specific RNAi drugs, what's the bigger picture for treating AATD-LD? Are there other approaches coming up?
Absolutely. The field is buzzing, actually. While RNAi focuses on silencing the gene, there's a lot of excitement around RNA editing.
Editing? How is that different?
Instead of just degrading the faulty mRNA, RNA editing aims to actually correct the mutation within the mRNA molecule itself.
Yeah.
Using enzymes, like ADAR enzymes, guided to the specific spot.
So you'd potentially turn the faulty Z-AAT instructions back into instructions for normal AT.
That's the goal. It could potentially restore normal AAT protein production while also reducing the toxic Z-AAT. Companies like Wave Life Sciences, CoroBio, Arana, they're all working on this. Wave has even shown some early clinical proof of mechanism data suggesting it's feasible.
Wow, that sounds potentially even more transformative if it pans out. Restoring the normal protein, too.
It could be. Then you have gene editing, like CRISPR-based approaches. Beam Therapeutics has Beam 302, aiming to make a permanent correction directly in the DNA sequence of liver cells.
A potential one-time cure.
That's the ultimate ambition, though it's earlier stage and carries its own set of complexities and considerations regarding permanent changes to the genome.
Sure. What else?
There's also traditional gene therapy, using viruses like AAV to deliver a correct copy of a serpin-1 gene. And people haven't given up entirely on small molecules, though, as we mentioned, Vertex had some recent setbacks trying to find chaperones to help the protein fold correctly.
Right.
And there are other biologics, like Inhibrx's INBRX-101 and therapies like alvelestat, still focused primarily on the lung disease. So it's a really diverse pipeline.
It really is. Lots of different angles. What are the biggest hurdles the whole field faces right now?
Several big ones. Demonstrating clear clinical benefit, especially fibrosis regression, remains key. That fazirsiran placebo response really highlights the need for robust long-term data and maybe better ways to measure fibrosis changes.
Yeah.
Long-term safety of any chronic therapy is always paramount. Navigating the regulatory pathways for these novel modalities, RNAi, editing, gene therapy, requires clear endpoints and dialogue with agencies.
And just finding the patients, right? AATD is often missed.
Hugely important. Improving diagnosis rates is critical if these therapies are to reach the people who need them. And finally, understanding the disease heterogeneity, why it affects people differently, and figuring out which therapy might be best for which patient profile.
Lots to tackle still. So, looking ahead, crystal ball time. What are the most exciting prospects?
Well, the potential approval of fazirsiran would be massive. It would be the first drug specifically targeting the liver disease mechanism in AATD. That would be a huge win for patients and really validate the RNAi approach.
A landmark moment.
Definitely. But beyond that, the potential of RNA editing and gene editing is incredibly exciting. If those technologies mature and prove safe and effective, they could offer truly corrective, maybe even curative options down the line.
Maybe combination therapies eventually, addressing liver and lung.
That's certainly a possibility in the future. And if these therapies work well in adults, exploring their use earlier, perhaps in pediatric patients with liver involvement, would be a logical next step.
This has been a really insightful look at a fast-moving field. It's genuinely impressive how targeted these RNA-based therapies are, getting right into the genetic instructions.
It really represents a significant step forward in precision medicine for genetic disorders. Fazirsiran, leading the charge with RNAi, offers real, tangible hope for slowing or stopping that relentless liver damage in ATD.
And maybe a final thought for our listeners to chew on. As these powerful genetic therapies become more refined and potentially more widespread, what are the broader implications? How might more routine genetic screening for conditions like AATD change healthcare, change individual lives, if we can identify risk much earlier?
That's a really big question, touching on ethics, access, and the future shape of preventative medicine. Definitely something worth pondering.
Indeed. Thanks for joining us on this deep dive.